Hello, welcome to our mutation lecture. Let me get my pen going as usual. Okay, so mutations are changes to the base sequence of a chromosome. So in other words, changes to the order or amount of bases, A's and T's and C's and G's in a chromosome. Mutations are caused by a number of things. First of all, random mistakes in DNA replication can be made by the enzyme that makes DNA, which is called DNA polymerase. Okay. Our next lecture will be about cell division, and in that we'll talk about DNA replication. Every time a cell divides, it needs to make new DNA, and DNA polymerase is pretty darn good at making a new copy of the DNA, to be exact, but there is a background rate of errors caused, and those are the mutations. It is thought to be that about 50 to 150 mutations occur per generation, but that number is controversial and it's hard to pin down, and some people disagree with that. But at any rate, there is just a background rate of mutations just caused by mistakes that DNA polymerase makes. Harmful chemicals can also cause mutations. Like, we all should know that cigarette smoking can cause harmful mutations in your lungs and give you lung cancer. And also radiation, sunlight or X-rays. The UV light in sunlight can give you skin cancer, and cancer is caused by a mutation. All right, so let's go through the different types of mutation. Okay. So there are several types of mutations. The first is called a nucleotide substitution. This is the most simple type of mutation. That's when one nucleotide is simply changed from one base to another. Right? This is also called a point mutation, and that's often what I call it, or sometimes just called a substitution mutation. So here we have our double-stranded DNA, and looking just at this base pair here, okay, it is AT, okay, and remember, one of these will be the template strand and one would be the non-template strand, okay. The template strand is what's going to be read to make the gene, and a substitution would be any change, right? So here we're just showing a substitution from A to T to A, but you could also change, say, the T to a C or a G. That would also be considered a substitution mutation, right? So these are flipped around, and this is still the template, and this would be the non-template. And what that means is whatever codon that A is a part of, it should have been a T before, okay? So if this is your codon, and now this is your codon, you're going to potentially get a change in the amino acid that's specified from it. Okay? So here's an analogy using a three-letter word sentence, or a sentence just composed of three-letter words. Okay, so the cat ate the rat, and that makes sense, right? And we're using three-letter words because that is how the ribosome reads the DNA, right? So the DNA, or reads the mRNA, which comes from the DNA. So everything's in three-letter words. A base pair substitution would be like if you took one of these letters, say the C, and you made it into an L instead. And now you have the lat ate the rat, which doesn't make sense, right? But you've only changed this one word from cat to lat, okay? So you haven't changed the meaning of a whole sentence, just that word. Sometimes base pair substitutions, oh, that's the other thing that I always call them, base pair substitutions. Sorry, these are analogous. Nucleotide substitution, base pair substitution, point mutation. Why are there so many words for one thing? I don't know, but there are. Anyways, base pair substitutions can sometimes cause no change if the codon that is specified does not specify a different amino acid. Okay. Another type of mutation are called insertion mutations, and this would be when in the DNA sequence one or more nucleotides are just added into the DNA, so they're pushed their way in there, okay, and that might take place in the middle of a gene, for example, or at the beginning of the gene, and it can have pretty dire consequences. Here's our analogy using three-letter words. The original sentence would be the cat ate the rat again. And what would happen if right here we added another letter? We added a G, okay? What that does is it changes the reading frame. Remember that the ribosome finds the start codon and that sets the reading frame, right? So the reading frame here is to read in sets of 3 words, or 3-letter words going down, right? And that makes it make sense. When you add in another letter, but you still read in 3-letter words, you've reset the reading frame. So now the only word that makes sense is the, and then we have a nonsensical word, nonsensical error. That's actually a word, but it doesn't make sense in this. And then a T just hanging out on its own, okay? So it makes the whole genetic message change, and that can be highly detrimental. And we see a similar thing with the deletion mutation. The deletion mutation is just the opposite of an insertion mutation. So rather than putting in one or more nucleotides, you take out one or more nucleotides. And we get a similar problem happening, okay? So if our original sentence is the cat ate the rat, then we delete something, say we delete this T here, okay? We've done the same thing, which is reset the reading frame, and we're still reading, excuse me, we're still reading in sets of 3 letters, but now we've reset it so we have CAA, because we don't have that T anymore, and now we have a nonsensical message again, okay? Both deletion and insertion mutations are called frameshift mutations. I don't think your book actually uses this term. But what it does is it shifts the reading frame, right? So the way that the nucleotides are set into sets of 3 changes if you take some out or put some in. Okay? And that can have a very big downstream consequence for the protein, because if you change the codons, then you're going to change the amino acids. Excuse me. Okay. And then there are some other types of mutations that don't involve just little parts of DNA, but big chunks of DNA, okay? And an inversion mutation is when a whole chunk of DNA breaks off, flips around, and then is put back right where it was before, but now everything's opposite of how it would be, okay? And again, remembering that one of these strands is going to be the template and one would be the non-template. Now you've switched everything, and that's going to mess up the codons again. There can also be translocation mutations, and I put in this diagram because I really did not like the one in your book. I thought it was incredibly confusing, and so I was guessing you would too. It's just a simple idea, really. Part of one chromosome pops off and is attached to a different chromosome, okay? And in this here, it's showing 2 ends of a chromosome being translocated, like they have exchanged their ends. Okay, and these are different chromosomes, chromosome 4 and chromosome 20. They contain genes for different traits. So this would really be mixing things up a bunch. Could happen in the middle of a gene and cause lots of problems. It can also happen that just this chunk comes off and attaches to this one, and no part of this one comes off. There doesn't have to be an exchange. And then finally, there can be a duplication mutation, and that's when a gene or a chunk of DNA is copied so that you then have 2 or more of them in the chromosome. Okay? And so that can add a bunch of genetic information. It can really mess up reading frames. It can also just make it so that there are multiple more copies of a protein being made. Okay, so here's a nice summary of the different mutation types. This does not have the nucleotide substitution, but it has the deletion, insertion, inversion. Oh, did we not talk about that one? Oh, no, inversion. We talked about that one, sorry. Translocation and duplication. Okay, I thought that was a nice little just, cartoony diagram to understand it. Okay, so we often think about mutations being harmful, and they definitely can be. There are definitely mutations, and we're going to go through one, that cause diseases. Genetic diseases that you inherit from your parents are often harmful, right? And they're caused by mutations. But mutations can also be neutral, like when I talked about the nucleotide substitution. If it's substituted to a nucleotide that still codes for the same amino acid, meaning it's still the same codon. Remember, the codon table has that redundancy built into it. Then you're going to end up with a neutral mutation. This would be no change to the protein. No change to the protein tertiary structure. Okay. Oh, and I should say, I meant to talk about this. So why would mutations be harmful? So remember, the DNA, that's where the change would happen, specifies the mRNA, which then specifies the order of amino acids in the protein, the primary structure of the protein, right? And then that goes and makes, you know, the tertiary structure of the protein, which then goes to determine the function. Okay, so if you change something in the DNA, you will see a downstream effect. You'll see a change in the mRNA, a change in the primary structure, a change in the tertiary structure, and then resulting in a change in function. So it could be a change in function, it could be a loss of function. Those things can cause diseases, but it can also be positive. So a neutral change would be that even though we've changed something in the DNA and the mRNA, when the protein is built, it still has the same sequence of amino acids. The primary structure hasn't been changed, and so then you would still have the same function. So not all mutations are harmful or neutral. Some are actually beneficial. Okay, so you could have a change in DNA that leads to a change in mRNA that leads to a change in the structure of the protein, and instead of making it not work at all or work more poorly, maybe it actually has a downstream effect of causing it to have the protein to have a different function or to have a better function, and that would be a beneficial mutation. Okay, so mutations generate new alleles, and alleles, oops, forgot about that. Let me erase this to get it out of the way. Okay. Okay, so alleles are different versions of the same gene. So here, this is a pea plant flower, and they both have flower color. The flower color is determined by a gene, and there are two different alleles. There could be purple flowers or white flowers, and so the alleles are the purple flower gene, or the purple flower allele, and the white flower allele for the flower color gene. Okay. So the way we get different versions of the same genes and why we don't all look the same is through mutation. So these generate alleles, which are different versions of the same gene. Okay? These increase genetic diversity within a population of organisms, and that's very important for evolution. Okay? And when we talk about evolution, we'll talk about the role of mutation and generation of new alleles. Okay? Right now we're going to go through an example of harmful mutation causing a disease, and then we're going to talk about some of the beneficial effects. All right, so remember sickle cell disease, SCD? We talked about that previously when we talked about protein structure. So sickle cell disease, go to this one, is caused by having hemoglobin molecules that are not the correct shape, right? So remember, hemoglobin has primary, secondary, tertiary, and quaternary structure. It has the alpha subunits and the beta subunits, okay, and they all have to come together to make a functional hemoglobin. And the hemoglobin, remember, carries oxygen around, delivers oxygen to the cells. Okay? So regular hemoglobin, normal hemoglobin, will be free floating. It'll be found in the red blood cells, but it won't be attached to each other. Sickle cell hemoglobin has the beta subunit. Those are the ones that are in pink, is mutated and has a funny tertiary structure that then causes these long fibers to form, and then that causes the cells to be sickled rather than to be nice and round. Okay, so we talked about that before, so if it's not ringing too many bells, you could go back to the protein structure video and watch that one again. Okay, so let's talk about the mutation. So this is a point mutation. Remember, that's a base pair substitution, aka nucleotide substitution. It's a change in a single base, okay? So it doesn't seem like that would be a big deal, but it can have dire consequences depending on where it happens and what kind of effect it has on the amino acid sequence, okay? So this is showing a bunch of information. This is showing the first part of the hemoglobin gene, and this is for normal hemoglobin, okay? So there's the non-template DNA sequence. This is in yellow, of course, where we're looking at the mutation. And then there's the template, right? The template is what's going to be read to make the mRNA, right? It'll be set into codons, starting at the start codon. Okay, so we're looking at this here. Okay. So in the template DNA, the codon should be CTC, and that would code for glutamic acid. Okay. Glutamic acid is hydrophilic. It's hydrophilic and it's acidic, right? So how is it going to interact when it's part of a protein? It's going to be hanging out on the outside of that protein, facing the water and interacting with water. Okay. In the sickle cell hemoglobin, instead of the DNA template being CTC, it's CAC. Okay, so we've swapped out a T for an A in the DNA, and that has the consequence of changing the mRNA codon to CUG instead of CAG, and that codes for an entirely different amino acid. It codes for valine. Okay? If you take a look at valine, valine has an R group that is what? That's right, very hydrophobic, right? It only has carbon, carbon, and carbon hydrogen bonds that are nonpolar. Okay, and so how does it act when it's part of a protein? It acts, by being trying to get away from the water, right? And so it's found in this region where it's pulling away from the water. Just like when you guys made the protein in lab, and all of your hydrophobic R groups tried to get, we're supposed to be in the center of your protein because they're trying to get away from the water, and that's what the valine does too. Okay? So a small change in the DNA results in actually a big change in the tertiary structure of that protein that then results in that disease where you have the sickled red blood cells as opposed to the nice round ones. They get stuck, and those small capillaries cut off blood circulation and cause all sorts of problems for the people that have them. All right, let's look at some beneficial situations. Okay, so here are some that have been studied that actually are mutations that lead to less disease and a longer life. Okay? So, there's a lot of people trying to figure out, like, why do some other people live so much longer? Is it their diet? Is it, their environment? Is it their genes? And yes, it's all of those things, but here are some situations where it's clearly a genetic difference, okay? All of these studies were looking at people who were genetically isolated. What that means is that there was not a lot of outbreeding. It was like a small group of people who kept reproducing with each other, okay? That can be very bad if there are detrimental or negative mutations. It can amplify them, and we see that when we look at things like we look at the one that hemophilia that causes people to bleed a lot, and we'll talk about that when we talk about sex-linked genes later in genetics. But it can also amplify positive mutations as well, and that's what happened here. Okay. So I'm just going to quickly go through this. So Ashkenazi Jews have, this is compared, so this percentage is compared to a control group, so people who are not in this isolated group of people. So they have genetic mutations that can decrease the amount of Alzheimer's disease, hypertension, which is high blood pressure, and diabetes, so they have less incidence of this. In Ecuadorian people, there's this thing called Laron syndrome, which actually causes people to be small, but it also causes them to have 100% resistance to both cancer and diabetes. So even though they are very small, they don't die of cancer and they don't get diabetes. Okay, old order Amish people, many of them carry this genetic mutation that infers resistance to cardiovascular disease, and then many Japanese Americans carry also a different mutation that confers cardiovascular disease resistance and cancer resistance. Okay, and then here's another one, an interesting case, talking about humans, which are Homo sapiens, and their skin and eye color. Okay, so to start off our discussion, just a reminder that humans evolved in Africa. Modern humans, the first modern humans evolved about 200,000 years ago, which is actually a very short amount of time on a Earth kind of time scale, where Earth has been around for 4.5 billion years. Anyway, so about 200,000 years ago, all humans lived in Africa, and everybody had dark-colored skin, dark-colored hair, and dark-colored eyes. Okay? There's a genetic component to this, or a reason why, and it has to do with this gene called the MC1R gene, okay, which is melanocortin 1 receptor gene. Okay? This is found on chromosome 16. What is this? This is called a karyotype. Okay? A karyotype is just a picture or a snapshot of all of the chromosomes found inside of a cell. We'll be talking about these more as we get into talking about cell division and talking about genetics. So for now, just know that humans have 22 types of chromosomes. We do have 2 of each. Types of what are called autosomes, just regular chromosomes that are not involved in sex determination. And then we have 2 sex chromosomes. Either you have an XX, an XY, or an XY. So this is a karyotype, and then I'm just using this to show that this gene is located on chromosome 16. Okay? This gene, MCR1, codes for a protein that's involved in regulating mammalian skin and hair color. Okay, so it's actually a gene that we find in all mammals that are involved in the skin and the hair color of all mammals, like dogs, cats, bears, all that kind of stuff. It codes for a membrane protein, so it means it's a protein that is stuck into the membrane in these specialized cells called melanocytes. Melanin is the pigment that is going to be controlled, and cyte is just a word for cell. So these are just like melanin-producing cells. Okay? These are specialized cells that produce the pigment melanin. Okay, so melanin, well, let's see. So MC1R controls the type of melanin produced. Okay? So there's 2 types of melanin. There's pheomelanin and there's eumelanin. Pheomelanin is red, and eumelanin is either brown or black. Okay? So when the MC1R gene is activated, that means it's turned on. It causes those melanocytes to switch from generating pheomelanin, which is the default, so all the cells start making just the red melanin. When MC1R is turned on, then they start making eumelanin, which is the dark colored one. Okay? So when you have a functional MCR1 gene, then you have dark skin and dark hair. Okay? So all humans started out with a functional MCR1 gene, and indeed, people who are indigenous to Africa and have, or from a population of, Africans who have lived there for a very long time, they all have the functional MCR1 gene. But it's not so when you look at humans across the globe. There's some differences. Okay? So eumelanin, or the brown or black one, acts as a natural sunscreen, so it's very beneficial in Africa where the sun is so harsh. Okay? So there's lots of UV light coming down, so it's important to have a natural sunscreen. Okay? But humans didn't stay in Africa. They started migrating out of Africa and first went up into Europe and into Asia. Okay? As they migrated, a new allele was generated, and that was an allele for red hair and fair skin. Okay? If this allele had been generated in Africa, it would be highly detrimental, meaning very bad. And it probably potentially was generated earlier, before people started migrating. It could have been, but it was not, like, it was a bad thing, right? But as humans moved further north, it wasn't so bad. Okay, so let's look at what's happening first. So fair skin and red hair first appeared around 50,000 years ago. Okay? and it results from a mutation to that MCR1 gene. Okay, so it's a mutation to that gene that activates the production of eumelanin. Okay, and this this mutation is known as V6OL allele. Okay. This V6OL allele, remember, an allele is just a different version of the gene. Okay, so this is a different version of that membrane protein. Okay, and this version codes for a non-functional MCR1 protein. Okay, that means that people who have this mutation don't have that MCR1 protein embedded in the membrane of their melanocytes. Okay, and that means that they never switched to producing eumelanin. They only ever produced pheomelanin, and that results in the phenotype, meaning what they look like, of having red hair and fair skin, because they don't make any eumelanin. Okay? In northern climates, light skin is not detrimental because the sun is not as harsh. Okay, and in fact, it can be very advantageous to have lighter skin because the melanin blocks sunlight, and that can prevent the absorption of vitamin D. Now, when the sun is super strong, that's not so important because there's enough sunlight to kind of, you know, meet those needs of vitamin D. But when the sun is less strong as you move farther north, and indeed like in the winter, there's very much less sun, then being able to make vitamin D from your skin becomes very important. Okay, so to make vitamin D, your skin has to be in the sun. And vitamin D is needed for in order to absorb calcium. So you can consume as much calcium as you want, but if you don't have vitamin D to help your body absorb it, then you won't get the calcium. And calcium promotes bone growth, right? Because our bones have calcium in them. So if you don't get enough vitamin D, then you get rickets, which is soft bones. So the bones don't develop properly in the children, and then that ends up with fragile, misshapen bones in adults. So this was an allele that proved to be a mutation that proved to be beneficial as humans moved further north. Okay, and we can look at some places on the globe, especially Ireland and Scotland and parts of England, and I think this is Wales, if my memory serves correctly, or maybe that's just Scotland. Anyways, we see a high percentage of the population carrying this allele, right? So a lot of people have it. So even though it's a mutation that caused a protein to be non-functional, it's still a beneficial one.